All right, thank you everyone for coming. And first off, a very happy Darwin Day to you. This is an International Day of Celebration, and I'm pasting in the about text from the Darwin Day website, and that we are celebrating really not just that Darwin helped revolutionize the thinking of nature and biology and evolution, but also trying to reflect on the bravery and the curiosity and the hunger for truth that his particular story represents from that. So I want to make sure that we recognize that what this day is about. And I am Stephen Gasior, Stephen Zoot Fly in Second Life, and I started this lecture series for the Science Circle, I think about 5 or 6 years, maybe 8 years ago, where I highlight the contribution to evolutionary thinking of some scientist that also in many cases also is characterized by this hunt for curiosity or breaking the mold or bravery in intellectual thinking. And I've selected quite a number. The criteria tend to evolve. Again, it has to be something that is really more of a specific contribution to evolutionary thought, or being a science communicator and proponent of evolutionary thought in addition to being a professional scientist. And so if you look through the back catalog of these talks, I've talked about Lynn Margulis, Barbara McClintock, E.O. Wilson, and Dawkins. And so this year I was really trying to find something that was a little bit unique compared to those. And what I'm really pleased to say is that something that I had just overlooked all these years is the contributions of Frances Arnold. And what is distinct from her contribution to evolutionary thought, and we'll cover all this and I'll be highlighting this aspect of it, is what evolution and understanding evolution can do for us today. Not so much a matter of understanding basic biological principles and the history of evolution and how the planet came to be, but really, in a sense, when evolution can contribute to engineering in the biological sense. So let's hit on it. So Frances Arnold, born in 1956, daughter of a nuclear physicist, so again, growing up in a household with a scientist already, and the granddaughter of a Army lieutenant general. She grew up in a suburb of Pittsburgh, and as a high schooler, she hitchhiked to Washington, D.C. to protest the Vietnam War and live on her own, and did various odd jobs. And that she also, well, not really enjoying, I think, school and not doing very well in it, she was actually clearly quite smart and demonstrated that on standardized tests and was able to attend Princeton University. She ultimately went on to earn a Ph.D. at University of California. Again, this was in engineering, and we'll come back to that in a second. Career-wise, she became a visiting associate at Caltech after having a very brief appointment at, I think, University of Minnesota. And that's where she ultimately has conducted her career, because you can see that she was an assistant professor of chemical engineering, then associate professor, then professor, and then finally she's actually leading, she is actually the director of the Rosen Bioengineering Center since 2013. And she has been recognized as the Linus Pauling Professor of Chemical Engineering, Bioengineering, and Biochemistry since the year 2000. And again, one of the big career highlights, and one of the reasons that she's gotten a lot more recognition in the recent years is that, and her work has gotten recognition, is that in 2018, she was co-awarded the Nobel Prize in Chemistry for the directed evolution of enzymes, which is going to be the basic crux of the talk for today. Yes, that's part of it, Phil. I will also say if you read her biography carefully, her father also helped, I think, get her name in there too. So that's definitely a part of it. And that's some other biographical tidbits that I want to mention is that, her father, again, considered quite a smart man within the nuclear physics area. He actually helped design the pressurized water reactor technology needed to make cheap nuclear power come true. Because, again, it's not just that you can generate heat, it's that you can also make sure you control the heat. Like I mentioned, she hitchhiked anti-war protests. Again, her starting at Princeton as an undergrad in engineering, that was at the very beginning of women even being allowed into undergraduate programs in engineering. Again, a reminder of the gender bias in education in the mid-1900s. Now, she was actually very interested. I think this is something you see from her biography. She's definitely someone who is enthusiastic about issues. She has a different way of thinking. She's adventurous. And what she was really excited about was the Carter administration's emphasis on clean, renewable energy sources, this whole sustainability. Again, before we called it sustainability. And she actually worked in that field in Colorado. But, like many other things that happen in politics, with the election of Ronald Reagan, the future for passive solar heating and cooling seemed somewhat limited. And so she decided to start graduate studies at University of California, Berkeley, where she specifically studied chemical engineering. Again, she didn't lose her interest in working in that field, but decided to pursue graduate school. Again, that's actually one of I think the famous parts of Reagan is, I think, like on the very first day, he had the solar panels that Carter had installed on the White House just removed. So I do want to make a quick note that while she was there, Alan Wilson was someone who introduced her to the molecular evolution of protein sequences. Again, giving her she's coming from a chemical engineering standpoint, but he was someone who's influential at giving her a little bit of this molecular biology perspective and background. And again, she went on to this deployment at University of Minnesota, but she met Jay Bailey, a world-renowned biochemical engineering professor at Caltech. And again, ultimately they got married, had children, and ultimately that's where they worked it out, where she started her assistant professorship career at Caltech. And so someone well set up to do some interesting things, Nobel Prize, paradigm shifting worthy. But before I get into her work, I want to make sure that, like I do for most Darwin Day talks, give some very basic background in terms of what the biology is. And then again, we'll be using this basic biology to understand the basic points of her contributions to the field without getting too much into detail. And so this here is a description, a pictorial, of the central dogma of molecular biology. Again, basically, the underlying principle of how we know cellular life works, which is you have a blueprint for genetic information in DNA, which is a double-stranded deoxynucleotide helix that gets transcribed by proteins into RNA, and typically that's only single-stranded in terms of functionality. And this code, this blueprint for the code, is then carried to a synthesis factory known as the ribosome, and then it's translated. So again, going from a nucleotide sequence into a peptide sequence. And that's what you're seeing here, is that enzymes would be that string of red balls that are coming off of the ribosome. And here is just a slight key point, is that on the right-hand side, there is a particular code that most life follows in terms of a set of 3 RNA nucleotides, ribonucleotides, that become a specific amino acid. And so this is known as a codon table. And what you see here on the right is that some string of UUU is phenylalanine, whereas a string of UUA is leucine. So again, this is the kind of Rosetta Stone of how we go from blueprint to actual enzyme. And one thing I'll point out here real quickly is that if you imagine being able to switch around from this basic code of U, A, G's, and C's, if you can just mutate that in the DNA, then you're actually ultimately changing the sequence of the peptide that's coming out of it. And so this is an important aspect of the process of really how easy it is to change the output of a gene in terms of these simplified bacterial systems that we'll be talking about for Francis Arnold's work. Yeah, and I think that, if, tagline mentions that, the structure of DNA was what we solved in 1953, and the Nobel Prize for that was 1959 for Watson and Crick. And then understanding the code was something that Crick contributed to. And, this is all work that basically molecular biology revolution only started in the 1960s. So again, this is the key part, and we're still learning about how protein folding works. Again, you might have just discovered that in the news, people talking about AlphaFold, that we're only now getting a decent handle on how to predict what protein structure looks like. And so that's my next slide, and that is, it's not that enzymes are just a bunch of amino acids as a bead on a string. They have to basically have a 3D form, a 3D topology that's very important. And what I'm showing on the left-hand slide here is this description of these, where you go from that original sequence, the primary structure, and then locally you get things that are called secondary structures. They fold into particular local structures, primarily alpha helices and beta sheets. One thing that's not mentioned here is that things that aren't forming into structure, things that are just kind of outside non-structured things, those are called loops. It's important to think of loops as also being places where things can change. It can be important. And the tertiary structure is when those alpha helices and beta sheets start to interact with each other and fold in a way that gives you its basic structure. And a quaternary is, in some cases, if you have a multi-protein complex, we talk about how those different proteins interact and come together in a very specific multi-protein structure. And it's important to recognize that the structure of the protein is important for how stable it is, how well it can interact with the surrounding solute, what structure it is, and then also how it interacts and grabs onto and then catalyzes reactions. And so that's what I'm showing up here in the upper right-hand corner, is that what you have on the left is an example of an enzyme. You see the little spirals, those are alpha helices. There's some beta sheets that you see a little bit hidden in the back. But the basic idea, and what's showing here, is that it can take maltose, again, a disaccharide that we get from our diet, and then it puts it into the pocket. You see those little colored structures in the middle there, the black, and it's surrounded by some red and yellow that are basically amino acid moieties in the enzyme. And then it converts it into glucose. So again, this is the idea. There's a substrate, and there's a product, and the enzyme catalyzes this reaction. As an example of this enzyme, this is something that would be happening with our intestines. So whatever sort of environment the intestine has would be relevant to how active it is. And then the bottom-hand side, just to understand a little bit more about catalysis, to understand that proteins aren't just these things that, when we look at pictures, we get the sense of a very fixed structure. And it's really important to recognize is that it's the flexibility of how these amino acids are in the structure and how they interact with the substrate that actually leads to catalysis. This is actually a very dynamic process, but is typically not very well represented in pictures. And so videos are a really good source for understanding this. But that's what I want to show here is that on the bottom right-hand side, showing this example of an enzyme where, again, the blue is the idea of a binding pocket, and then with absent the substrates it's got a particular shape. Then once the substrates are interacting with it, you'll notice that the shape on the bottom is much more closed. It's trying to bring basically different chemistry together in order to then usually cause some sort of stress in the system that then causes a catalytic chemical reaction. And so that is kind of the basics of understanding how enzymes get made, what they do, and again, the important parts is that the structure, the sequence, and the catalysis are things that are all related to that basic primary structure and function and the string of amino acids and how they interact with substrates, how they interact to form a structure. And one thing to recognize, and that this is kind of intuitively obvious, is that these have certain optimal conditions in which they work. And so this is a diagram. What you see on the x-axis is just temperature. And because they're scientists who made this picture, it's in Celsius, not this Fahrenheit nonsense. And then on the y-axis, you're seeing so from higher as you go up the y-axis. And what you can see is that at different temperatures, you have different amounts of enzyme activity until you reach this optimal temperature, in this case about 47 degrees. And then typically what happens is, if you keep things, well, what inevitably happens for any given enzyme, the hotter you get, more is that the temperature can also affect the catalytic site. And so this is one thing to keep in mind, is that usually these things are evolved in a way that's most important for when and where they're acting. Now, temperature, which is something that's imparting energy into the system, into the structure, sometimes helping unfold it. There are other aspects too. And so the pH. Now, I'll use the term pH, but just keep in mind that that's just a description of how much positive or negative charge you have within the solution. And that's what I'm showing here on the left, is that if you imagine that there are a bunch of positive charges that are interacting with that left-hand face of the enzyme, that those are going to interact with the positive and negative moieties that are on the enzyme. That can change their structure, their location in space as they interact. But you can also imagine on the right-hand side, if you allow negative charges, that any given enzyme, as it interacts with that negativity, can actually change structure, function, the ability to catalyze reactions. And yeah, so tagline puts it in slightly more complicated terms, which is the environment in which proteins or enzymes function is potentially a cause of disruption of the folded tertiary structure on which function is dependent. And I think that's a very nicely scientific way of describing it. Another example is also the aqueousness of the solvent. So what I have on the right-hand side is, again, the same enzyme, and those little red and white balls, those are water. So again, a lot of things we talk about in biology are around a water, aqueous environment, where there's a slight bit of negative and positive charge. But what you have on the right-hand side, that is DMF, which is a organic solvent. And so the fact that it has those three little white balls connected to a black ball, that is a methyl group, and that ends up being very nonpolar. That's more organic. And so those moieties, again, as that interacts with the surface and the different amino acids of an enzyme, that can dramatically change how they're folding, how they're interacting with each other, and whatnot. So again, if you imagine one thing you do when you wash dishes, when it comes to getting rid of a lot of oily or greasy food on your plates, then you use soap, which has this ability to be both charged as well as provides fatty chains that helps dissolve things that are more fatty. And so I think when you put that kind of together, this is an example. That's an example of how things can interact with the substrate. Okay, so with that kind of basic understanding, we will get into Arnold's work. So this is the first paper that came out in 1991, published in Science. And what is being represented here is subtilisin E, is this enzyme. It's basically a protease. It's something that looks at proteins and chops them up in some way. And when you have this in a bacterial colony on a plate, and you add a bunch of protein around, in this case casein, again, a large component, say, of milk, then you can actually visually see halos, that this enzyme gets secreted and degrades it in a way where you have this obvious example of how active it is. I do want to say another thing is that a lot of the work that you see here is described as the specific, looking at a very specific substrate where you can measure the rate of reaction, which is just this sRPNA, and that's how I'll describe it. But basically has some amino acids on a sugar that can be degraded and then quantified. And what's shown here is a diagram from the picture. And there are a lot of details that are sitting around it, but I just want to come back to the main idea, which is what they're doing is using a technique known as PCR, which is a way to amplify DNA, which of course is involved in putting in those DNA sequences, but they're doing it in a way that's error-prone. And so that DNA sequence, that string of A, G, C's, and T's, will have occasional errors in it that can lead to a new and novel protein sequence. And so, again, this is that contribution from Alan that helped out with her way of conceptualizing, doing, again, chemical engineering. And again, to be true to the table, again, don't try and analyze and understand all this. I will walk you through it. But what the table is showing at the top is comparing the wild-type enzyme, and we can just highlight the wild-type enzyme and the bottom one, which is a combination of 3 mutations that are designated as O. We now have a Q that's being substituted with an R, or a D being substituted with an N, or an N being substituted with an S at these various positions. And this one column where I've highlighted the orange is showing that as you add this organic solvent, DMF, that the wild-type enzyme has a certain catalytic efficiency. Again, we won't go through all the details of the catalysis and what else means, but just think of catalytic efficiency, how well it takes a substrate and turns it into a product, is 6.5 units for the wild-type. But when you add these 3 mutants, it basically becomes 110. So it becomes a much higher catalytic efficiency in a completely different environment. And then as they go to an even higher percentage of dimethylformamide, the 20%, then you see the wild type is getting worse because, again, it's an enzyme that's adapted for aqueous environments. And the mutated version is actually, again, still extremely good and even relatively much better at 45 units. And so, again, these random mutations that they sorted through and looked for within the colonies, they found them, from this visual assay of casein digestion, and then specifically characterized their specific activity with the substrate. On the lower left-hand side, again, what we have on the x-axis is the concentration of DMF, and on the y-axis, going higher, is that catalytic efficiency. And you can see that all across these different concentrations of DMF, and even in an original aqueous solution, the mutant is just much better in terms of catalytic efficiency than the wild type. And so, and that's what we also see on the right-hand side is, again, what is some of the contributions of this, is the idea that if you take an enzyme and put it into dimethylformamide, in this case 40%, it's going to denature, right? Instead of it being surrounded by happy oxygens, it's getting surrounded by an organic solvent, it'll start to denature. And again, the right-hand bottom diagram is showing how this mutant version is able to survive that denaturation better than the wild type. So the next figure is from the Nobel Prize website, again, one of the best diagrams I've seen for this, which is explaining what this is set up and the basic idea of what the Nobel Prize was awarded for. And the idea here is you start with DNA, and you introduce in some fashion or another random mutations. And then these mutations are used to express the enzyme in some sort of system, in this case using bacteria. You have to clone it back into the bacteria to get it expressed and to look for that phenotype. And part 3 is an example of how, like in this case, the colonies on agar would have halos of casein that are being digested. And then basically, through some sort of quantitative, or in this case, kind of semi-quantitative way, they're just observing which ones are better than the rest. And so again, that would be a single round of directed evolution. That is your evolution from variability of the sequence, then evolution, which is a selective process that you set up in your assay. And then this is the big key part here, is that you just keep iterating on this in a very rapid fashion over and over with fresh sets of mutations and then fresh sets of assays. And so if you feel like you've understood this basic concept, then you have understood the basics of this Nobel Prize. And that the value of this is that for any given enzyme that we're really interested in trying to get activity out of, this is an incredible evolution in fast motion, accelerated way that we can just keep improving whatever that enzyme is as well. So any kind of quick questions on that? Because if you're confused here, I'm going to be, I myself, most of this talk will be kind of iterating on this basic concept over and over. So now is a good time to kind of pause if there are any questions. Okay, I will then get one. So with this idea here is that in this first paper they did a few rounds of selection and PCR, and then basically they gave them a new version of the enzyme that was better. And then they had a series of papers, basically still working on substilicin E. And again, I won't go through this one very much, but this sets up the stage where they then took that variant they had and then iterated on top of it. And so they went through more rounds of mutagenesis, and they kept pushing the platform of how much dimethylformamide they were starting and comparing. So now that once you have something that works pretty well in 10% dimethylformamide, then you actually mutagenize that and then keep testing other versions of that in a higher concentration of dimethylformamide as much as you can to then keep iterating. And so from that version from last time where we had 3 amino acid substitutions, they now got to, well, they actually added a 4th. They got an additional 6. And so the bottom line here is that this PC3 substilicin is 130 times more efficient than wild type in now 40% DMF. So again, they were really pushing the percentage of DMF that they had. And that's what you see in the right-hand diagram is just, again, this nice example of over a wide range of conditions that, especially if you compare to the previous one, this PC3 version, now with, again, a total of 10 mutations, is just so much more catalytically efficient. And then I think the trilogy of papers here kind of ends with this one. And this is where I also put in a nice diagram picture of this, which is what I've been showing before is this idea of catalytic efficiency. But in any given reaction, if you're trying to get a product out, what you really want is better total activity. You really want to make sure that you're getting the most of your compound, your product as you can, which is slightly different than catalytic efficiency. And so what we have in this table up here, and what the experiment was, is comparing that wild-type version of the gene and then the 10 mutant version that we just talked about last time, and they took it through several more rounds of mutagenesis, again, with an eye on total activity, not just efficiency, but total activity. And that's what you see here in this table that I have circled in orange, is that once you got to 20% DMF, then you're getting a dramatically larger amount of total product from all these multiple rounds of mutagenesis. Again, as an example, in terms of showing catalytic efficiency, again, keep maintaining increasing catalytic efficiency in the graph you have on the left-hand side. And what's really important, so this is a 3D model of the protein, and it's showing the green represents the structure, the ribbon of it, and then the yellow are where the mutations that were introduced into it, where they reside. And I think this is the other flip side of understanding how the power and impact of this biology is that not only are we trying to make something that works better. And it doesn't really, we don't have to know how to make it better. We can basically do all sorts of randomness in order to generate these mutations. But that once you have them, if you can go back and understand the structure, function, relationship of where they are, you actually start getting insight into how that enzyme's working in the first place. And so this is a classic technique of introducing mutations to understand how something works when you make it worse or make it better. And this is, again, putting this into a bit of an overdrive, whereas you start finding as many of these mutations as you can in multiple cases, then you start getting insight to how it works in the first place. But it's also a pretty neat and colorful picture. All right, so a lot of what I've talked about is trying to look at increasing the catalytic efficiency, which again also has to do with its protein stability in an organic solvent. And this paper that came out in 1999 is again looking at the same enzyme, substilicin E, but trying to make it something that works at a higher temperature. And so that's what we have here on the left-hand side, is trying to show and develop a half-life. Again, how long is its half-life at a higher temperature compared to the starting point, which is 0, and then the first round of mutagenesis, second, third, fourth, fifth, sixth round of mutagenesis. And now what you actually see at this first round of mutagenesis, they found 5 different mutations that all individually contributed to being more stable at a higher temperature. And I'm going to come back to this in the next slide, but what they did to make that second generation was they actually went back and combined, through another bit of technology, how to basically take those mutations. They didn't do more mutagenesis. They just tried to find the best combination of those 5 different mutations to give them the platform to then do more random mutagenesis. But what you see here, again, this is nicely demonstrating this principle that every generation of mutagenesis you're attempting, you just keep selecting for another one that's better and better. Now again, there are variants they found here that were obviously worse, but as you keep selecting ones that are better and better, you keep improving multifold how much better it is. Yeah, Sumo brings up that point, is that it's like breeding, it's like artificial selection with, say, domesticated animals. You just keep selecting the ones, just keep grabbing that, keep getting the best ones, and then ultimately, if you keep using that as your as the platform for your next generation, it just keeps getting better and better. Now what's shown here on the right-hand side, this is, again, just like that thermostability graph I showed before that was theoretical. This is demonstrating exactly for these genes, or for these different enzymes, their profile of activity over different temperatures. So again, the x-axis is temperature from 0 to higher, again in Celsius. The y-axis is activity. That the little line, that's the bottom one, that's wild-type substilicin. The dark circles is the mutated version that they ultimately came up with, the best one they came up with. And then the gray dot line, again, the one that has the most activity, that is an example, and that's what they're talking about, is thermatase. That is the same, it's a homolog, it's basically an enzyme that's very similar, but has been living in a thermophile, basically a bacterium that has evolved to survive at high temperatures. So, of course, its natural activity over evolution is to be very stable at high temperatures and high catalytic activity at high temperatures. But what this is demonstrating is that through this mutation strategy, you can evolve an enzyme to be similar to that relatively quickly. And that's actually a little note I have here at the bottom from the paper, is that the thermatase is actually different from substilicin E at 157 different amino acid positions. But this evolution strategy, this directed evolution, they only needed 8 amino acids to get something, somewhat equivalent. Okay. Here is, again, some more kind of eye candy in terms of 3D enzyme structures, but this is demonstrating, again, a space delay model. So it actually is including all the amino acid moieties, the atoms that are present in there. And then space delaying in, the blue are showing where these mutations were made, and the gray is showing the substrate. And so this is interesting, right, that in order to be more thermostable and to catalyze this reaction better at a higher temperature, you're, in many cases, making new substitutions in both the active site, the places that are binding the substrate, but then also it's really important to do this on the surface of the protein. There are places that are not near the busy part where all the action is in terms of catalysis. It's actually in the overall structure of the enzyme. So Noah Wonder asks, is directed evolution the right term for this? And if you're talking about this as in Francis Arnold's contributions, then yes. And that's because you're always setting up an assay that you have a specific quantitative way of picking the ones that are better each time. It's directed towards some sort of specific activity you're selecting for. Now, in nature, if you're kind of referring to the commentary in local chat, then not really, right? In evolution, you do have different selective pressures, selecting those by design in order to find the best ones. It kind of happens because, but what are the best selective pressures are not necessarily known in nature. And of course, it changes over time. So I do want to come back to one thing, and this is kind of a parallel contribution being made by other researchers, is that remember how I mentioned from that first generation of mutations in subsalicin, they combined them together to get the best one from combining those different mutations. And this is a technology that was invented in 1994 by a researcher named Stemmer. And what he showed in a test tube, that instead of using PCR to randomly add in specific mutations at bases, what he did would take different versions of the same gene. And what that's being depicted here is, say, in evolution, there's an ancestral gene which has diversified into different species and different versions of it by the different colors, is that you can do this thing called shuffling, also known as molecular breeding, in a test tube, that you're not introducing mutations, but you're just trying to make different combinations of them. And that's why you see this mosaic of colors in the chimeric genes at the bottom. And what's really powerful about this technique is that, it doesn't have as much variability possible from random mutagenesis. You're actually combining a bunch of different genes that are all functional. And so that way the output is that you're not accidentally introducing mutation that's creating a completely non-functional version of your enzyme. And what's really trying to look to do is that if these things are evolving in different species and are trying to find different versions that work maybe a little bit better for their situation, then putting those together in different combinations can give you something that's actually that much better in the end. And so this is called DNA shuffling. Also, some people call it molecular breeding. A lot of people think that Stemmer could have been co-awarded the Nobel Prize in 2018, but unfortunately he had passed away in 2013. So again, dead people are not eligible for the Nobel Prize. But again, also a very powerful technique that adds to this toolbox of mutagenesis. And this is where I want to kind of step back and give a little bit of a conceptual way of thinking about this overall biology and technology. And this is from a review by Francis Arnold. And what you're seeing on the left is some examples of techniques you can use to take an enzyme that maybe isn't very functional for what you want to do. Just starting with some sort of minimal activity, or maybe some baseline activity that you want to improve. And that as you think about the theoretical space you're working with, this is kind of a 3D conceptualization, is what you have on the right. What you have on the z-axis getting higher is better activity or better fitness. And then you have different sequence, the x and y axes are kind of representing the different kind of sequence space you have for different substitutions. And you can think that in theory, there's a peak. There's one particular sequence that is just the very best representation of this in theory. And so that's what these mountain ranges are showing, is that as you look at a different variety of sequences, theoretically there's some peak. And what you do in all these technologies with random mutagenesis or with DNA shuffling is you're just trying to start from a dot, like you have the red, the green, and the blue dot that's at the base of each of these like mountain-like looking things. And then you're trying to explore the space to basically keep slowly crawling up the peak. Now, in some cases, you see in the blue dot, it can be the case that you go exploring up one peak, and then because there's a valley between the two, you actually can never get to that optimum. You just might never quite get to that because you followed a certain branch path. But the green is kind of the theoretical example you really kind of want to see, which is you start with low activity, and you keep incrementally changing and basically climbing up that mountain step at a time until you get to a much better enzyme. And so this is kind of that a way of conceptualizing what this technique is doing is trying to basically incrementally get to a better spot with just exploring space. Okay, so next. Now, Francis, I mean, basically that's the foundational work and basically started the entire field of directed evolution, or what's sometimes called molecular artificial selection. And that the promise, as you can see from a basic standpoint, is making any enzyme just much better, or maybe even being better at different substrates and what sort of chemistry is produced. And so I want to give an example of a few other highlights from our work. This one is, looking at a different enzyme, notice cytochrome P450. Again, I think it was Syzygy was saying he was expecting to see hemoglobin later. We're not going to have hemoglobin in this talk, but what characterizes the cytochromes is that they do have a heme in the middle, or maybe it's tagline. Maybe it's tagline he's talking about expecting hemoglobin to come up. But cytochromes are these enzymes that produce and have a lot of different capabilities in biology. And that's why I wrote a quick summary up on the right-hand side saying, these typically are doing oxygen metabolism, adding oxygens to groups or shifting them around. They have a wide variety of functionality in mammals. They will oxidize steroids, fatty acids, xenobiotics. They're important for like drug metabolism and stuff in the liver, as well as hormone synthesis and breakdown. In plants, they've also been seen to be important for defensive compounds, fatty acids, and hormones. And of course, within the bacteria, they're doing a wide variety of things that are relevant to the environment of the bacteria. And what Arnold is doing in this paper, and what you see on the left-hand side, is that this compound, naphthalene, which is being presented to the cell, it can, by default, perform a particular chemistry on it, turning it into adding an oxygen to the first or second carbon. And that's actually what you see on the right-hand side. Sorry, let me go back. What they also engineer into the cell is horseradish peroxidase, that when that metabolized compound then is interacting with horseradish peroxidase, it will make a different fluorescent compound. It basically adds it on to a platform fluorescent compound. And that's what they're showing on the right-hand side, is that if you look at column 1, you see column 1 is a bunch of green. That if you have just this default cytochrome, the wild-type P450, it makes green. But in setting up this assay, when they start mutating the cytochrome P450, then you actually have it adding different intensities. Its reaction rate is better, so it can change the intensity of the colors. Can also change where the oxygen is going. And when it changes where the oxygen is going, then you get a different color after horseradish peroxidase adds it on to the fluorescent polymer. And so that's what you see on the right-hand side, is starting from column 3 all the way through column 11, are all these different varieties and mutations in P450 that are changing its basic chemistry, changing the chemistry of what product it's making from the same substrate into different compounds. And so this is a way of, and what's really powerful here, what's really important here to recognize is that this is the beginning of high throughput colorimetric assays that allow this to happen at a much higher rate. It's not just that people look at colonies and look at halos anymore. People are trying to use, high throughput machines that are just, say, looking at colors, or maybe doing some other sort of digital recognition of variation in order to do this at a very high rate. So an important contribution. Another highlight from her work is, again, taking a look at another cytochrome, where what the cytochrome typically does is converts a fatty acid that has a double bond carbon at the end into a unsaturated, a single bond. And so that's what you see on the left-hand side, is that for lauric acid or palmitic acid, the wild type, the black, has a certain reaction rate, a turnover rate. And then they mutagenized that enzyme in order to, and what they saw was an increase in the rate that happens. But what I want to point out on the left-hand side is that while the bottom two are biological molecules, that is what it does in the chemistry of the cell. Octane, hexane, cyclohexane, pentane, butane, propane. What do those sound like to you? Are those biological molecules? Yeah, they sound like tanes. I'd say thank you, Neuro, for that one. Yeah, these are basically petroleum products, gasoline, hydrocarbons. Again, as a chemical class, they're hydrocarbons, and we use them for a variety of chemical synthesis properties. Again, propane is an example of a heating element. And so the ability to catalyze the basic chemistry of those hydrocarbons and then, again, add an oxygen to them to basically oxidize them is something that these enzymes couldn't do. But if you look at the gray bars from the mutation of the mutated version of the cytochrome, you see that they get a dramatic increase amount of activity. And so that's kind of the bottom line on the right-hand side, is that when it comes to the substrate hexane, so again, hexane, 6, circular molecule, hydrocarbon, that compared to every known naturally working enzyme that can catalyze that into hexanol, again, an alcohol version of hexane, so adding a hydroxyl group, that this P450 variant they've created is way better than anything that's found in nature. And so this type of engineering is an example that in terms of something that is really not exactly the favorite substrate, can actually create a huge amount of difference in activity. And again, I just want to show in the next thing, this is a review from, not Arnold, from somebody, from Arnold, talking about why cytochromes end up being really interesting places to do directed evolution. Because they actually have several catalytic steps, they have the ability to bind different substrates and work pretty well. Now, again, I did read an actual industrial review, and that cytochromes end up having a lot of difficulty in terms of getting them to work at really high scale. And in terms of thinking about gigantic vats of these doing some sort of chemistry, it's a little bit more difficult than that. But as an example of a place and a platform to work on understanding and to okay, so this is where, again, I'm just borrowing from the Nobel Pages website to highlight. Again, I think I wanted to give some of the science that gives the platform for understanding what Francis Arnold's early contributions to the field were in terms of the basic science. I'm putting together some summary here that from the Nobel Prizes, literature. Again, these slides will be on the Science Circle's website as well as my works cited, so there'll be a note card that if you want to peruse all these resources, that'll all be available. But the pharmaceutical world and the sustainability, the environmental remediation spaces are where people think about using these chemical tools of life, enzymes, in order to do these reactions we need that are unnatural. And so trying to basically convert natural enzymes to deal with our unnatural byproducts and substrates. And that's really where her contribution of learning to designing new enzymes to optimize their activity, to give them new types of activities, could fundamentally change the chemistry, applied chemistry. Her research group developed enzymes that can transform simple sugars to isobutanol. So that's an example of something where in a high fermenter space, you can basically create a high energy compound that's very useful for energy distribution, but is also a relatively sustainable organic way to produce it, including plastics. Also working on, again, cytochrome P450, for which there were no other enzyme available before. That's the example of the paper I just talked about. They also found a cytochrome variant. Again, they actually changed the absorption spectrum of it, so that's where the numbers come from, is the absorption spectrum. And they found variants that perform, say, an azide reduction more efficiently than nitrogen transfer to sulfide. And so that way they could create these that create the specific process. And again, one thing that's kind of cool is I think, for those of you who know what enantiomers are, enzymes, you can design them in a way that they're always producing the right enantiomer of your product, whereas a lot of times in chemical reactions, you get a mixture of the two. They also were able to get beta carotenoid production. That's, again, vitamin A, which you get from carrots. They actually took that pathway, which is found in plants and yeast, and actually got it to work in E. coli and optimize it. So again, taking the biological chemistry of one species and optimizing it into a much easier to work with organism. And then other valuable chemicals like L-methionine. L-methionine is a really important food product or animal feed, and be able to produce that at high levels. And again, in summary, what the Nobel Prize Committee was saying is that directed evolution of enzymes has become a highly efficient protocol for the development of biocatalysts with high specificity, limited side reactions, and tolerance of diverse reaction conditions. And again, this is a summary, a relatively recent review that I got, which I think does a nice job of capturing up to date what some of the applications of this work are. That, again, CO2 capture and utilization, very important for trying to extract extra carbon in the atmosphere. If you know much about methanogens, these are enzymes or these are bacteria that can basically pull carbon from an inorganic source into an organic source, and maybe they can do that scale and make those reactions much more efficient. Bioremediation of toxic pollutants. Again, if you have an oil spill, there's lots of strange chemistry in there, a lot of strange molecules, trying to get that broken down, whereas the environment normally has trouble handling that. Deconstruction of plastics. So that's actually a chemical reaction I'm showing here at this next stage, is that polyethylene terephthalate, PET, that's an example of a repeating unit in plastics. Again, something that, again, the problem with plastics is that they're not biologically derived, and so there aren't very many natural examples of chemistry to break them down in the environment. So plastics accumulate; they don't get degraded, and so this reaction here is an engineered version of an enzyme that actually is able to catalyze and break that down into subcomponents, again, including ethylene glycol. So again, trying to think about, again, if we have industry that creates problems, can we use industry to help solve those problems? And then renewable feedstocks is another good example where, again, can we, at high scale, produce food or other important biological compounds that people want to eat from the chemistry of from the chemistry of bacteria. And this is actually a really good example. If you've heard about lots of meatless burgers, this technique and applications are being applied to that as well. Now, revolutions beget revolutions. And this is something that I'm familiar with from my own work. And again, I won't go through all the details. There's just some key points here, which is, in the upper left-hand side, again, DNA, molecular biology, relies upon people being able to make novel sequences of DNA. Okay, spell correct, change it from DNA olives. It's supposed to be DNA oligos. That's oligos. I'm just noticing that now. And then a lot of times we'll use those building blocks of small DNA oligos into larger sequences. And what's this graph is showing here is that back in 1963, they figured out how to do this, and then they invented a relatively efficient phosphoramidase chemistry to do that. And then they discovered that enzymes can do it, but it's really not until 2021 where now the application of this technology, it becomes relatively easy to make DNA that's 10 kilobases in size. So again, scaling up the ability to make synthetic DNA. Again, just making DNA completely in a test tube from an enzymatic reaction. And so that scale of going from really just a few hundred base pairs at best 10 kb is something that's going to usher in that next revolution of microbiology. And what's responsible, the connection to Arnold, is that these accomplishments recently is because of basically directed evolution on an enzyme known as terminal deoxynucleotide transferase, or TDT. And it's a polymerase that basically is able to just add DNA onto the end of an oligo, and you can control the chemistry of the reaction to make it specifically sequence you want. And this used to be very inefficient. This is something that hardly worked and made lots of errors back in the early 2000s. But with this directed evolution strategy, we are now at this point where we're making 10 kbs a sequence. And there's actually a whole bunch of papers, a whole bunch of companies that are now marketing this. And so I think, again, while microbiology has already gone through several revolutions of scale and what we can do, from someone who's in the field, this is going to be another big stepwise change to what we can do and can do. And so, in summary, and that's an example. Again, there are other examples in other fields where if you're more of a specialist, you might know some of the more interesting things going on beyond what I had in the last two slides. But for this contribution, to understand how to use the principles of evolution in order to engineer a heightened, better, faster chemistry of enzymes, then Francis Arnold is my 2024 hero of evolution. So thank you for your time. Thank you for your attention. If you have any questions, I'm happy to answer those. And again, like I said before, the resources for this will be available on the Science Circle website hopefully soon. Thank you.